analysis, we need to now compare all of the sequences. So we've uploaded our data, we've done the blast to find what our particular fish is. So now we need to select the data that we're going to use in the final analysis. So here's the data that the reference data that we uploaded, and here's the blast hits that we selected from the results of the blast search. So we need to save those. And now we need to compare them. And the way we do that is a program called multiple sequence alignment. And the specific one is muscle. So we're going to click that to run. And this is going to give us an alignment of all the sequences put together. It takes a few minutes for muscle to run, and now it's done. So we can view the results. And here's why we call it a DNA barcode. So we have where you see different color bars means that there's a sequence difference. And where you have gray, that means that the sequence is identical. Okay, so now if you see with drawing the straight lines, it looks like a UPC symbol you might find at the grocery store. And then here's our sequences. These are dark because they haven't started yet. Okay, so they start here. And so you can see that our sequence and the ones from the database line up very closely with our reference sequence here because they have this difference, whereas none of the other fish share that difference at that position. Okay, you can see these fish cluster in their cod, and you see they have differences that aren't shared with the other fish. So this is how we start to look at the relationships. Then, and you can see here more differences that are shared with the tilapias and our fish, but they aren't shared necessarily with these other groupings, the tunas here, the salmon, and the cods. Okay, so that's the sequence. Another way to look at that is to look at the sequence as letters. And so again, what we're seeing is that there are differences, and they're highlighted in reds. Okay, and that if it's gray, then the sequence is identical. So what we're just looking at is the differences, because it's these differences that allow us to determine the relationships between all the fish, and also to confirm our comparison that we think that this is a type of tilapia, which I will look up. We'll look up the species name in a moment, but previously I know from previous work. Now what we're going to do though from mussel, it's interesting, but it's kind of hard to see the exact relationships. So what we do is we turn it into a phylogenetic tree. And I like the one that's called Philip with ML for maximum likelihood. Okay, these are just two different ways of inferring trees, and I like this one, so I'm going to use maximum likelihood. Okay, and now here's our tree. So, so now you can see just by glancing at it, even if you're not familiar with looking at trees, that these fishes are grouping out in small, what are called clades. And you can see the cods grouped together, all the tunas here. Okay, here's our fish, and it's grouping with tilapia. Okay, coming from here to this clade. This is called a node, and so anything descending from a node is a clade. This is also a clade, so you have to be specific when you're talking about nodes and clades. And then here the salmons grouped together. And then here we have what's called the outgroup, and this helps us to determine these trees and get the relationships correctly. So this is a cartilaginous fish, right? Related to sharks and stingrays, whereas these are all bony fish. So this is a very distant related organism to the ones that we're actually interested in, and it helps us, helps these programs, the software, draw the tree correctly. Now, so here's our grouping, and we know that we can see both from the multiple sequence alignment as well as now here that our particular fish is most closely related to tilapia, because we only have to go back to this node to get to tilapia, okay? And its next most closely related grouping is the tuna, okay? And we know that because we only have to go from here to get to tuna, whereas to get to salmon, we have to go from this node to this node to that node, so we got to go deep into evolutionary history in order to now come to the sockeye and to the salmons. So you determine the relationships from trees not by reading from top to bottom. That has no meaning. It's by counting back nodes. So you have to learn to read trees by looking at node position. So you can't say, well, it's more closely related to this, to tuna, just because it's next to it in vertical order. It's because of the node counts. Okay, so it looks like we have identified a tilapia. We're going to now go look and see specifically. And again, from our blast analysis, there were a couple of differences from this one than to this one. And this shows that this is identical because this little branch right there means that it has some differences, sequence differences. These are identical. These have just very, very few differences. So the sequences are very closely related. Whereas these two, which are both catfish, they're very distantly related catfish, but they're more closely related to each other than they are to the salmons. Okay, so the branch length here tells us something about how close or how much sequence difference there is between the two species, and also something about its evolutionary relationship. So now we need to go and learn more about these different organisms. So what you can do is download your tree for future use. What I would also suggest, though, is once all members of your group have finished the analysis, is that you upload all of the sequences that you analyzed here and generate one big tree. So make sure you select the data again and rerun muscle, rerun the tree building, and you'll have all of your species on the same tree. And you can see how they're related to each other. Okay, so now we want to learn something about the organisms, and we will come back to that in just a moment.